today is to show a little bit about the techniques we use and we developed in our labs to address protein oligomerization in the live plasma membrane. So as a motivation, I choose one of the proteins we're currently working on. So during my talk, you can exchange this protein by any other membrane protein. So this is what I explained today is not restricted to this particular protein I will talk now. But this is a showcase protein I use for the motivation. And this is one of the proteins which now, I think keeps us busy in more than 10 years. And this is the T cell receptor signaling complex. So basically T cells are part of our immune system, and T cells that detect antigen in our body. So I always like the image of a T cell being a little tiny computer, and the main task of the computer is to discriminate between something bad and something good. So it discriminates between self-peptides and foreign peptides. And this process of detection, you can hear on the left side an electron micrograph of a T cell in red, interacting with an antigen-presenting cell, is a very dynamic process, as you can see on the right side. So these are lattice light sheet microscopy recordings of a T cell, so in red, interacting with an antigen-presenting cell. And in this case, it's even a cytotoxic T cell. So the process ends by the T cell killing the antigen-presenting cell. But you can see that the process is highly dynamic. So the membrane is fluctuating. You have a lot of membrane ruffles. The actin cytoskeleton is extremely dynamic. So a lot of things are going on at the same time. So the protein responsible for detecting now, or discriminating between good and bad, it's a T cell receptor, CD3 signaling complex, which is here in the central image, this protein complex. And it detects the antigen, which is presented in the context of peptide MHC molecules on the antigen-presenting cell side. And we know, so let's zoom in a little bit more into this complex. It's a multimeric complex. So basically, it's an octameric complex of different subunits, and the alpha-beta subunit being responsible for recognizing the antigen. We also know from our own studies that this process is incredibly specific and also sensitive. So we know that even a single antigenic peptide on an antigen-presenting cell is sufficient to activate a T cell. The process is also very specific because if this is a self-peptide, T cell should not activate. Some people are suffering from autoimmune disease or allergy. So for these cases, this process is not working properly. But for healthy people, the sensitivity and specificity is given. But the main thing here is that we don't know why the T cell is so sensitive. Why is a single antigen sufficient to activate the T cell? And we wanted to start really right at the very scratch to answering this question. So we wanted to know how does the T cell receptor look like on the live cell plasma membrane? So what is the stoichiometry of the T cell receptor complex? Because the image I've shown you here, this is the typical image you see in textbooks. But there have been some biochemical data which suggested that for this complex, there might be several antigen-binding subunits, alpha-beta subunits per complex. So basically, several subunits can interact with the peptide and thereby increasing the signal, amplify the signal, finally leading to T cell activation. So we wanted to image this T cell receptor on the plasma membrane. And I would like to show you how we did it. So first of all, because of this highly dynamic system, we decided to get rid of the antigen-presenting cell and replace it by a two-dimensional model system. In this case, we used supported lipid bilayer membranes prepared on cover slips, which carries a certain amount of lipids carrying nickel and Ta. And whenever you have nickel and Ta in the bilayer, you can add histidine-tagged proteins to the bilayer, which can bind on the surface, and thereby you can functionalize your membrane. So we use different proteins for binding. So for example, we used ICAM1, which is responsible for the adhesion of T cells. We can add co-stimulatory molecules like B7-1. We can even add the activating molecule, the peptide MSC class 2 molecule, and thereby we generate our own antigen-presenting cells. So we generate an APC mimicry. And the big advantage is that we control all parameters here. Because we can control the density of the proteins, we can control the type of proteins, and we can also choose the color of labeling, whatever we want. So this is the model system for replacing the antigen-presenting cell and to make it two-dimensional. Now the cells are missing, the T cells are missing. And for this particular project, we worked with primary cells. So we worked with primary cells from transgenic mice, where we exactly know which antigen is recognized by the T cell receptor. So every T cell receptor in this mouse T cell is the same, and it recognizes a particular antigen we know. So we can study the process of activation rather easily. So it's easy to get this cell. It takes maybe a week or 9 days for preparation, and then you have a couple of millions of T cells. You can just add to your supported lipid bilayers, and you can see in white light, you see a nice interaction between T cells and the APC surrogates. Of course, if you want to do single molecule fluorescence microscopy, you need a labeling of the T cell receptor. And in our case, because we want to know something about the stoichiometry, we want to know something about the oligomerisation, we also want to label the T cell receptor in a stoichiometric way, which means we would like to put exactly one fluorescent marker per T cell receptor subunit. In this case, we'll first talk about the beta subunit. So it's one part of the antigen recognizing subunit. And we wanted to target the beta subunit with the fluorescent dye. So how do you put now in a very specific one-to-one stoichiometry a dye to a protein? So I think maybe all of you are aware of different options how to label proteins on a cell membrane. So in this case, we have several options. So one of the, let's say, simplest ones is to buy an antibody, label the antibody with fluorescence dye, and then use it to bind, in this case, to bind this epsilon subunit here, this green part. However, the antibody has the big disadvantage, it's huge. So it's 150 kilodalton, it's really huge. And also for T cells, it's a no-go because the antibody has 2 binding epitopes, and as soon as you cross-link 2 T cell receptors, you immediately get T cell activation. So already just trace amounts of antibody are sufficient to activate the T cell. So this does not work. The other options you have, we can use an FAB, so a fragment antigen binding, which is just one part, about 1/3 of the full antibody. It's much smaller, and you can also label it unspecifically with fluorescent dyes. This is one option. And we'll talk about this a little bit later. However, our chosen tags normally for these studies are either genetic tags, so you can add an GFP, for example, any fluorescent protein, or another tag where you can, like a halo tag, SNAP tag, where you can later covalently bind the organic dye to it. Already with a much smaller size. What we choose for this study was an engineered single-chain variable fragment. And this basically is just the outer part of the antibody linked together, because normally the 2 chains are not linked at this site. So it's an engineered mini antibody, which can also be site-specifically labeled. So you can, for example, add a free cysteine, and then you can label every single-chain fragment with one dye molecule. And what we also like to work with are nanobodies. So nanobodies nowadays are available for a lot of different binding targets. They're really small, just 15 kD. They can be easily expressed in bacteria, for example, so you can have huge amounts, and you can also site-specifically label them with a fluorescent dye of your choice. So we more or less, for the techniques I will describe later, prefer these strategies for labeling. And for the particular case of the T cell receptor, we use the single-chain fragment. So you can see here on the bottom right, the molecular structure. So you see the T cell receptor in green, the peptide MHC, which is in the antigen-presenting cell, on the bilayer in our case, in magenta, in cyan. And on the right, in red, you see the single-chain fragment. And the single-chain fragment has several advantages. First is it's small. We can add a dye at a certain position of interest. And also this single-chain fragment does not interfere at all with the binding of the peptide MHC molecule, which was important for this study. So this was in collaboration with Johannes Huber. And yeah, so it took our collaborators 7 years to really design the perfect single-chain fragment. So sometimes it can be really, really tricky and long-lasting to find the perfect labeling strategy. But in this case, we had a nearly one-to-one binding stoichiometry. So every label, so every single chain carried exactly one dye-molecule with 99% efficiency. And because of the tiny size, we could also target nearly all T-cell receptors on the T-cell membrane. You can tune also your imaging later by choosing the dye of your choice. So for this study, we mostly use the Lexapro dyes for several reasons, which I go into detail in a few minutes. Okay, so far I introduced the model system. So we have this antigen-presenting cell mimicry. We have the T cells. Now we have labeled the beta subunit of the T cell receptor. Now we need some imaging. And for imaging, we choose TIRF microscope. Why we use TIRF? So total internal reflection fluorescence microscopy, because it gives us a really nice contrast when looking at single molecules, because we just excite the synapse, so the interaction between the T cell and our bilayer. So just this tiny layer here is excited. So to be a little bit more flexible, so we built our microscopes in our lab because we're a physics lab. So we built the microscopes on our own, and just to tell you one of the simpler setups we are using, how it is constructed, and it might not differ from the setups you've seen or using on your own. So for the excitation, this TIRF microscope we use nowadays mostly laser combiner. So different lasers are built into a box and are coupled out via fiber coupling. So we have different wavelength in the box, and the laser is guided by a variable mirrors into the backboard of the microscope, and finally then into the objective. So the lasers we are normally using, because we're doing some bleaching approaches I show you later, normally have a laser power of around larger than 150 milliwatts, which give us around 3 to 10 kilowatts per square centimeter in the object plane. So this is an important value for us later then. We do some beam shaping. First of all, we would like to change the laser beam. So we have a little telescope, and we also need to focus the laser into the back focal plane to get a nice parallel beam out of the objective. We also can add some beam homogenizers to go away from the Gaussian shape of the laser beam, but to have a flat profile. We also do some other beam shaping, which means we just add in a conjugated plane, we add some adjustable slit, and thereby we can adjust the region which is illuminated on our sample very precisely. And here's the nice thing, because we're a technical university and we get a lot of students from physics courses and classes, and they're very interested in building their own microcontroller-based hardware. We have this nice motorized apertures, which can be controlled by a little microcontroller, and it's very convenient because you just have to press a button and you can change the field of view with not spending more than, I think, 200 euros, in addition to buying this mechanical aperture. We also use them for detection. It's important to have an high-NA objective if you do work with single molecules. And also for TIRF, we need objectives because it's objective-biased TIRF illumination we do. So we need objectives with an A's larger than 1.45 and 60 or 100x oil immersion objectives. To make life also more simple, we nowadays introduce some active focus stabilization systems, so so-called focus hold system. You can buy them commercially, or you build your own one, which is not super tricky. So basically a piezo under the objective controls the position of the objective, and whenever your image goes out of focus, it is automatically refocused by the focus hold system. So I don't want to go into detail, but it's very convenient to have such a system. And in addition, we need to see single molecules. We need a very sensitive detection system. So I will show you later some examples where we image single GFP molecules. And if you want to image a single GFP molecule, you don't get too many photons out of a GFP. So you want to have sensitive cameras here with a large quantum yield. And in our hand, either EM-CCD cameras work really well, or for special cases, we still use old liquid nitrogen cooled CCD cameras, which have been introduced, I don't know, a couple of 20, 30 years ago for astronomy, but they're also perfectly suited for single molecule, very low light application, single molecule microscopy, where you basically don't have introduce a lot of noise to the readout process. So I really like these cameras a lot, although I'm not really comfortable to handle. And the last thing, because we also do multicolor imaging, we build our own image splitters. I will also tell you later why it's important to have a good image splitter available. And fortunately, we also get, as I mentioned before, good students, and even one of our image splitter now was put into a product by a student. So basically, he built his own image splitter, and we're very happy with that. Okay, so now we have introduced all the tools. Now let's put the T-cells onto the model system. And what you see here is a white light image of a T-cell. So it's around 5 to 7 microns in diameter. This is a standard white light image. And I will switch immediately to fluorescence, and you will see then the labeled T-cell receptors. In this case, we labeled the beta subunit of the T-cell receptor with an Alexa fluo dye. And yeah, well, so the T-cell settles. So you can see at the very beginning, you see microvilli touching the supported lipid bilayer membrane, and then the T-cell spreads out. So it first touches the membrane, and then it spreads out and builds a huge, nice two-dimensional interaction plane. However, you can, just looking at this image, you don't see a lot of details. So you see some inhomogeneities in the signal, but basically you see there's a bright signal all over the cell membrane. So if you do a little bit in deeper analysis, you immediately find out that the density of the T-cell receptor is around 70 T-cell receptors per square micrometer. If you do just simple calculations, so 70 TCRs per micrometer gives you a next neighbor distance of 50 to 100 nanometers, which is of course much smaller than the diffraction limit of our microscope. So we're using white light lasers in the visible range. So the diffraction limit is around 250 nanometers. So it's impossible to see single receptor entities here because the density is too light. And here comes now the technique we use now since nearly 2 decades. And I would like to guide you through this technique, which we call TOXL. And TOXL stands for thinning out clusters while conserving the stoichiometry of labeling. And this is very simple. And you immediately will recognize that it's a FRAP approach. Yeah? So fluorescence recovery after photobleaching is a very famous approach to study membrane proteins, for example. And this is rather similar, but it's on a single molecule level. So what you see here in the first image is, again, we call it the pre-bleach image. So this is the image you've seen before. You have a high density of molecules. You don't see any details. But what you also see is just a tiny part of the cell. So some part of the cell is shielded by the aperture. So we close the aperture to only illuminate a part of the cell. And also the illumination is done in turf microscopy. So you only really see the bottom membrane of the T cell. And if we now zoom in into one individual camera pixel, we have no idea. So we know that there are a lot of T cell receptors, but we don't know, is it T cell receptor? Is it a monomeric T cell receptor? Is it maybe forming dimers or even higher oligomers? We don't know at this point. So the idea now was to turn on the laser, and to turn on the laser to around 10 kilowatts per square centimeter for half a second, and to photo bleach all the molecules you've seen on the left side, to fully photo bleach them. So to photo bleach every individual molecule. And you can check for that because this image here was recorded directly after photo bleaching and you see nothing. Yeah. So it's scaled differently. So there's no single molecule left in this area. But now if we wait, so this is the high power bleach pulse, but now if we wait for a couple of seconds, in this case, we waited between 5 and 10 seconds, we see single of these receptor entities now coming back. So due to the Brownian motion, all the T cell receptors are in motion. And some of them, which were in the shielded region, which have never seen the laser, and also coming from the top of the cell coming down into the field of view, they are now visible as diffraction limited spots. Yeah. And if you would wait another 10 seconds, you will maybe get the same image at the very beginning. So then the density is too high again. You cannot resolve single molecules anymore. But right at this onset of this recovery process, you see single signals. And now what you can do is you can analyze the brightness. Yeah. So if you have a sensitive camera, you can analyze the brightness of these individual dots. And based on the brightness, you can distinguish, is it 1 diamolecule per diffraction limited spot? Is it 2 diamolecules? Or is it more diamolecules per diffraction limited spot? And calculate back to the number of molecules. And this works quite well if your camera works in the linear range. and you have a sensitive detection system. So to show you what comes out, so in a typical Toxel experiment, you collect hundreds of these images, not just a single one, but you can collect hundreds of these images from different cells. And what you basically calculate is a histogram. You calculate a histogram of brightness values, and this is shown here in this diagram. So on the x-axis you see the brightness. Here it's a probability density function in this case, but you can see it as a normalized histogram. And the black curve is the brightness distribution which was measured. Now let's do a control experiment. So for analyzing this data, we need a monomer. We need the brightness distribution of a pure monomer signal. And you can do it in different ways. So one option is to just underlabel the T cell, that just a few T cell receptors really carry a label. You can also bleach the cell unstoichiometrically, just turn on the laser, wait a couple of seconds, and then only a few signals will remain. And most of the signals which will remain will be single signals. You can calculate based on probability, but it's easy to get this monomer control. You can find different ways for doing it. And you can also calculate the intensity distribution of these single dye molecules. And this is not, it's hardly visible. This would be the blue line. And now you do a very simple fit. So you assume that the data, which is described by the black curve, which is this probability density function, is just described by a linear combination of monomer contribution, dimer contribution, and so on. There's a linear combination of different contributions, and you can easily do this, but just by looking at the diagram, you already see, okay, our data can be explained by purely monomers. So there's hardly a dimeric fraction, which is this dotted line here. So basically the fit yields, it's more or less pure monomers. But now what happens if we look at a different molecule? What happens now if we look not at the beta subunit, but what happens if we look at the epsilon subunit? Because the T cell receptor carries complex, like in the textbook, carries 2 epsilon subunits. And if you label now the epsilon subunit with a different single chain fragment, but you can use the same dye molecule and repeat the same experiment, you see the data looks completely different. Yeah, so it's much broader, the brightness distribution. It cannot be explained now by only a monomeric contribution. It can be described by 25% of monomers and 75% of dimers. So this is basically the simple mathematical readout of this data. So for the beta subunit, we hardly see dimers, 3%, but you see the error is rather high. And for the epsilon subunit, we see nearly 3/4 of the signal which recover carry 2 dye molecules. So more or less, we could prove that the textbook's images are right. And we don't have a huge contribution of higher oligomers of the T cell receptor here. And before I will maybe tell you more about the 3%, whether they are really important or not, I would like to draw your attention on the 74, because we really try to be perfect here. So we really try to have everything perfectly prepared, the model system, the labeling. So we were somehow surprised to end up with 74 and not with 100%. Yeah, so in an ideal world, you will end up with 100%. So the question was, why do we get 74 and not 100%? And this is where we decided to go to computer experiments, so do in silico Toxel experiments. One of the reasons was also the COVID-19, because some students could not access the lab anymore, and we tried to find some projects which could be also done from home. So the huge advantage was that we know all the experimental parameters, because we did already all these experiments I've shown you before. So we know all the bleaching times, recovery times, so we know all the settings from the experiments. And we know also all the intrinsic parameters of our cellular system. So we know how fast is the T cell receptor moving. So we know diffusion constant. We know the surface density, the cell size. We did bleaching curves of the dye. So we know everything about our system. So we can take all this knowledge and put into a computer simulation. And this is what the result of the computer simulation looked like. So what we simulated, so we tried to simulate something like you've seen before for the T cell receptor. We simulate a certain density of molecules, in this case 100 molecules per square micrometer, and assumed 100% of dimer. So let's assume. So in the view from before, you can say, okay, this would be labeling of the epsilon subunit. No? So every molecule carries 2 dye molecules, and every red dot here corresponds to a T cell receptor carrying 2 dye molecules. So when we simulate the photobleaching and take a snapshot directly after the photobleaching process, so we have recovery time of 0, you can maybe see already from this image the appearance of blue dots. And a blue dot now means it's a partially photobleached dimer. So what basically happened is that during the bleaching process, only 1 out of the 2 molecules was photobleached. Yeah. So toxel, like I've described before, in the name of the technique, we have conversation of stoichiometry of labeling. And obviously, this is not absolutely true, because here we change the stoichiometry of labeling. Because in an ideal world, we would either destroy both fluorophores or non-fluorophore. But if we just destroy one, it looks like we have monomers in our system, although there was a pure dimeric system simulated. Yes, and if you look after a longer recovery time, like after a second roughly, you see that the molecules go into the central field of view, and you can also see that maybe the number of blue molecules decreases because they are intermixing again with the red ones, which come from outside. The blue ones go everywhere. So basically, you see that the number of blue dots decreases, but you can also quantify it, of course. So you can. We just did exactly the same quantification like we do normally for cells. So we just select the very central area here of the simulated area, and then we just count how many molecules still carry 2 dyes and how many are just partially photobleached. So now you could ask, why do we have this partial photobleaching? What are the reasons for that? So we could see 2 things for this partial photobleaching. The first thing is that molecules are diffusing. So what can happen is that molecules diffuse from the outside into the center during the bleaching time, but they also go out again. So maybe a dimer diffuses in during the bleach pulse, only 1 molecule is photobleached, and then it diffuses out again. And at a later time, we detect a partially photobleached dimer. The second region for the appearance of these blue dots for these partially photobleached dimers is the bleaching profile, because also the bleaching profile is not perfect. So again, we try to restrict the field of view by using an aperture, but the aperture edges are not perfectly sharp. We also have diffraction at the aperture edges, so we have a certain intensity decay at the edge, and this is symbolized here by this gray and black shaded line. So we have a certain area where the intensity from max value of the laser goes to 0, and it's not very steep, extremely steep. And that's why also the bleaching efficiency in this border region is pretty poor. And this is the second reason why we generate this partially photobleached diamonds. It's also worth to note that these are the 2 points which can lead to the appearance of monomers, although they are not here in the system. However, we can also have the opposite problem, that suddenly higher oligomers appear, although we did not simulate them. And this will be the case if 2 signals are too close together. If they're closer together than the diffraction limit, we will detect them as only a single signal. And for example, like you see here you see a dimer and a partially bleached dimer, but if they're too close together, we just see in a microscopy experiment a single fluorescent spot. And if we calculate the brightness later on, it will look like there are 3 molecules, 3 dye molecules on there, because we cannot resolve them. So this also we have to consider for the analysis. So what we did basically is we did a lot of this Monte Carlo simulations, and also to make it not too boring, just show you the most important outcome of these. So we fixed the bleaching time. So we bleached our system, and then we were at the recovery time. And just look at the gray line, which shows you the amount of dimers. So what you've maybe also seen before, and when I presented you this image, is if you look at directly after photobleaching, you could see a lot of blue dots, which means blue dots is referred to as a false monomer. So you have a high fraction of monomers. But the longer the recovery time is, the more intermixing happens, and the more red signals come back into the central field of view. So we found a clear dependence on the recovery time, and from that we can say, okay, at around 2.5 seconds of recovery, our dimer fraction is roughly 70%. And we see also monomers which are not true monomers. This is the error we make here just with using this technique. So this was the first finding. So it depends on the recovery and on the photobleaching time. But more interestingly, it also depends on the diffusion coefficient, because I also told you before, the molecules will move in and out of the photo bleach area, maybe during the bleaching process. And if it diffuses slower, the probability is lower that it comes in and out again and in maybe at a later time point. So what we did here, we varied the diffusion coefficient. And you see if the molecule is diffusing very slowly, we detect a high dimer fraction, but the faster it moves, the lower gets the dimer fraction. This makes absolutely sense because the faster it moves, the higher the probability can move in during bleaching and out also again. So these were all interesting findings for us to understand the technique we are applying on a microscope a little bit better. What really opened our eyes was the following: when we assume that dimers, that there exist monomers and dimers on the cell membrane, and they diffuse with a different diffusion coefficient. And this makes absolutely sense. So assume you look at a protein, a monomeric protein, and if you have the protein may be dimerizing by whatever means, then the protein is larger. And the larger protein aggregate will move slower in the plasma membrane. And this is what we also try to simulate here. So we simulated now a system where we see 50% of monomers and 50% of dimers. And if both of them diffuse with the same diffusion coefficient, so this is what you see here, is this diffusion coefficient of dimers versus the diffusion coefficient of monomers. And if both of them diffuse with the same diffusion coefficient, we see that, we detect around 50% of dimers. And now just please look at for the ideal laser profile, just at the dashed line. So if they diffuse with the same speed, we should detect ideally 50-50. However, if the dimer is a little bit larger and diffuses just with half of the diffusion coefficient, we immediately end up with completely different values. So we end up with maybe 13% of dimers, although we simulated 50%. And the reason is that the faster molecules, they will populate first the field of view, and the slower molecules will come later, but we are heavily biased towards the faster ones. And so this was important for us to also see the influence of different diffusion constants of monomers and dimers. Okay, so let's come back to the example of the T-cell receptor before, what I've shown you. So what we basically did, so we know all the details about the T-cell receptor. We know surface density, diffusion coefficient, we know the experimental parameters. So we can correct for this apparent dimer fraction we observed. So, and the correction easily yielded a corrected dimer fraction of close to 100%. So this outcome of the experiment of 75% was truly just because of the generation of partially photobleached dimers, and the Q number is around 100%. So this is what we could show. But still, I would like to go back to the brightness analysis part of before. So we know we can correct for the apparent dimer fraction, but the question is, can we also increase the sensitivity a bit of this Toxler experiment? And the answer is yes. So we can do the whole thing I've shown you before in two colors. So you can label the T-cell receptor, for example, half of it with a green dye, half of it with a red dye. You need an image splitter then, so you see the same part of the cell in green and red. You do the photo bleaching in both colors. You do a control image after the photo bleaching that you photo bleach both channels perfectly. And then after the recovery happened, you take an image with both color channels, and now you see red and green dots recovery. So for the two-color experiment, you can do also the brightness analysis, but to be more sensitive, you do a localization analysis. So you localize the single molecules in these images, and after localizing the single molecules, you look for co-localized signals. So, and here it's very important. So you need to really perfectly align the two color channels. And the image splitter is not sufficient for good alignment. So you need some fiducials to really align the two color channels and do a mathematical correction and see whether you get red and green signals co-localizing within a certain distance in this field of view. The reason why it's still very important to have a good beam splitter is to be perfectly in focus in both images. And this is what we also found out to be a very critical point. If you have low light applications, to have both images perfectly in focus. And either you can build your own image splitter, so you can use commercial ones and using some correction lenses, but still is not perfect. And with our own image splitters, we were able to really finely tune the focus and to get both images perfectly in focus. Okay, why is the two-color localization now sometimes better than the one-color brightness approach? Because it can also be used to detect extremely rare colocalization events. So here we show you, so I will go now away from the T-cell receptor for 1 second, because here we did a different test system, because we used some lipid body by gene 1 in the bilayer, and labeled it with cholera-toxin beta subunit, which was in red labeled, and we just used a very low colocalization probability. So basically we had just a few, we had a high amount of, I think, cholera-toxin subunits. So the colocalization probability was really low, but still we could detect colocalization events down to smaller than 2%. So this is important if you just have a few oligomers maybe in your cell and you still want like to detect them. But then you also have to think about the false positive rate, because what also can happen is that they're just random colocalizations. And in order to get rid of random colocalization, you can combine this two-color toxel approach with tracking. So after you do the recovery, you can do a tracking, so which means you just record images with a high time resolution. And if the two molecules are not only colocalized, but they also move together for a certain amount of time, then this is even more proof that you really have a true colocalization events. And this is what we exactly did then for the T-cell receptor complex. So to do these two-color toxel experiments, and just to show you the results here, this was now again done for the beta subunit. We labeled with Alexa 647 and Alexa 488 dyes. And if you look for colocalization events, and again, many cells, different cells, we ended up with colocalization of around 0.4%. So we cannot exclude that these are true dimers, but the question is whether they have really biological relevance if it's 0.4%, but we cannot exclude. If we do again the control of the epsilon subunit, we see that we detect around 25% of colocalization events, which is even higher if we do a lot of corrections, which I don't want to go into detail here. So for maybe the remaining 5 minutes or so, I would like to go away now from this particular example of the T-cell receptor and show you some other examples. So which may be also interesting for you to use this simpling bleaching approach for other proteins. So that's why I would like to just show you what we did so far. So everything basically started with looking at a completely different model system. So I started, and I have to say this was my master thesis a long time ago, where we looked at a model protein, a GP, linked via GPI anchor to the outer leaflet of the plasma membrane. So we looked at GPI, GP expressed by CHO cells. And because 20 years ago, looking at lipid clustering or like lipid rafts was quite popular. And so we also joined this field. And what we basically detected using this toxel technique was that GPI-GP is not traveling alone in the plasma membrane. Indeed, if we do this toxel approach, and that's why I mentioned also before this liquid nitrogen cooled camera, because single GP molecules, as you know, they are not really bright. You need sensitive systems to see every single molecule. And what we found out basically is that 30% of all GPI-GP molecules are forming homodimers. So they're somehow linked together in a cholesterol-dependent fashion, which was quite interesting at this time. And what you could also see is that this depended on temperature. So when you increase the temperature, so here's shown the dimer fraction. So at room at 37 degrees, it was around 32%. If you increase the temperature, this structure dissolved, and we could link to the expression of heat shock protein. Yeah, so this worked for basically a lipid anchored protein quite well. For a completely different project, we looked at a receptor tyrosine kinase, in this case ERP3, from an image here, which can form homodimers or also heterodimers with ERP2. And we used a different labeling approach here because we didn't have any single chain fragments or nanobodies available. So we wanted to use antibody fragments for this project. But just to maybe tell you in 1 minute what we run into problems when using antibody fragments. First of all, what we found out that the purification of FAPs is very critical. If you have used kits to dissect full antibodies, you never get rid of all antibodies. And we found out that maybe one antibody, 1 out of 1 million FABs, and you have one antibody still functional, this can change everything and can cause a huge bias in your experiments. What our collaborator also knows is because you want to get rid of all these parts. And if you label the FAB with organic dyes, also the number of dyes is very important, because if you have too many dyes, then maybe the affinity suffers from the antibody. If you have not sufficient dyes, then also you have an unlabeled dyes, and it's really hard to conserve the stoichiometry for labeling. So using FABs is really critical, but it still works if you want to use Toxel. And because of time reasons, I think, skip the details, but we could use also the FABs for 2-color experiments, and we could, again, so you see the labeled B3 in this case, and you do a colocalization analysis after the bleaching process, and we could see a clear homo-oligomerisation, which was much larger when we stimulated the cells with a ligand. Also here I would like to point out, it was really important to make control and correct for the degree of labeling and also for the diffusion coefficient, because the stimulated receptors diffuse differently than the unstimulated ones. Okay, with that, I just added this slide. So if you're interested in this technique and you want to know what we did maybe up to now and look for proteins, so this is a little choice of proteins we basically did in our lab. So I just have we looked at some molecules were at the inner leaflet of the plasma membrane, like LCK and T cells. We looked at ion channels, we looked at the serotonin or dopamine transporter, and also at the peptide MHC class 2 molecule. So we looked at very different proteins using Toxel, and more or less got successful results out of it. Okay. With that, I would like to make just one slide conclusion. So from the T cells, because this is how I start, and I would also like to end for the T cell receptors, we know that the monomer T cell receptors are the one which interact with the peptide MHC and which lead to final T cell activation. There are no T cell receptor oligomers in the very initial signaling process involved. We also think that supplementing your experimental data, or our experimental data, with Monte Carlo simulation really helps to understand better how the experimental system works. And you can also use it for correcting your results and also for finding the optimal experimental parameters. So you can also use these experiments first and then go to the microscopy experiments. And the take-home message hopefully for you is you can use this bleaching approach rather nicely to determine the stoichiometry of, and now that's the most important point, mobile membrane proteins, because the proteins need to be mobile, because everything we need a recovery after photobleaching. And with that, I would like to just thank most of the students in blue here and the images who did most of the work I presented to you today. Our whole biophysics group, the collaborators, so the T cell receptor work was all done together with Johannes. Felix from Aprima, who developed now this nice image splitter we are using, and also thanks to Baubi, who introduced me also to this community and made it available